Hello, friends. My name is Dr. Rajeev Kumar Kapoor, and I'm here for the Faculty Development Program, which is being organized by PSGR Krishnamal College for Women. As you know, that the Faculty Development Program is focused on enhancement of knowledge and skills through scientific literature. So when it comes to enhancement, uh, it is expected that we all know what is going on around us. So we need to enhance our skills so that we can impart the latest and up-to-date knowledge to our students. And since this faculty development program is organized by the Department of Botany, I appreciate the organizing members, especially Kanchana Madam, who invited me for this inaugural talk. Now, I am not a botanist, but I am a microbiologist. And today, the world is faced by such a huge challenge that is of the COVID virus, that it becomes very imperative for all the scientific people coming together and fight against this novel challenge which is being posed by this novel coronavirus. And that's the reason that a microbiologist has been invited so that we together uh, come together and work together for fighting against this novel coronavirus. So my topic of my talk is to provide you with the research updates when it comes to what is the research update and the vaccines and the treatment and in the diagnosis of this novel coronavirus. So in my talk today, I'll be briefing you about the latest findings for the in terms of the available technologies which are there for making the vaccines and the kind of therapies which are available for treatment of this novel coronavirus. So with your permission, I share my screen. You can see the COVID-19. Actually, that is the recent challenge for the entire mankind, uh, since a lot of lives have been lost due to this COVID-19, and we are here as a team of scientists who are working towards finding solutions. So we do need botanists, we do need medical instrumentation people, and we do need people from all disciplines of sciences to come together and find solutions against this deadly virus. My presentation is broken down into 4 different sections. I'll start with a brief introduction of COVID-19. Then I will present with an update on what are the available vaccines, what phase the vaccine has entered, and what is the recent report from the scientists all around the world. After that, I'll be presenting an update on the available therapies which are there, though there are no fixed therapies, but still, whatever is available for the treatment of this virus, I'll be sharing with you. At last, I'll be discussing the technologies available for the diagnosis of the virus. In this picture, you see this is what the novel coronavirus looks like. This is an electron micrograph because the size of a virus is in nanometers, and that is not even visible by a normal compound microscope. So this images shows you that how the virus looks like. You can see that it's the spike proteins projecting out of it, and this is the one which has posed such a big challenge for the mankind. Now, when we talk about coronavirus, actually, coronavirus is a group of viruses. It is a family of viruses. And COVID-19 is the latest member of this family of viruses. So this coronaviruses are a large family of viruses which are known for causing illness in across the different groups of animals, starting from the canines, the bovines, and even the dogs, and even the birds. This virus is known to infect all those creatures which are present in and around us. So, amongst the whole lot of the coronaviruses, there are 7 groups of viruses which are known to infect the human beings. And these are further classified into alpha coronavirus and the beta coronavirus. And if you see that the beta coronavirus, which recently in the year 2003 and 2012, there were 2 outbreaks of these coronaviruses. One is the MERS and other is the SARS. So these 2 outbreaks have been recorded previously in the history. The COVID-19, what we are facing today, is the recent member, or it is the new member which has been isolated for the first time from the Wuhan city. So the COVID-19 is actually an infectious disease caused by the recently discovered virus, which was isolated from the animal market of Wuhan. This new virus and disease were unknown before the outbreak. This was the first time that such an outbreak with this novel strain of virus has occurred. So this happened in China in the month of December 2019, and now it has become a global pandemic. That means it has spread across the continent of our globe and has affected each and every country, because all the countries were connected through the airlines and through the ship route, and the humans were the carrier of this virus. Let's talk about the symptoms. The early symptoms of the virus may start appearing within 1 to 14 days. Now, this is something which is really concerning, that a person who is infected by this virus may start showing the symptoms between 1 to 14 days. So, an infected person might not know that he or she is infected with the virus, because the symptoms will take between 1 to 14 days to appear, with a minimum of 6 days. Now, the early symptoms you can see here in the slide is the headache, which is a severe headache, running nose with extreme of sneezing, and a dry cough and sore throat is the initial feeling what you get after you get infected with this virus. Then you feel extremely tired, and you always find yourself short of breath. There is some difficulty in taking breaths. So, and which might later, after a due course of time, result in some complications, like you might suffer from a fever, a high fever. You might suffer from short breathness and acute pneumonia, hemoptysis, or severe diarrhea. These are the later symptoms which, once you develop the complete infection inside your body. Now, the transmission of the coronavirus has been traced down to the animals. There are many infections, many viruses which are there moving in different animals. And the proximity of humans to these animals, which are actually reservoirs of these viruses and many more diseases, causes infections to occur. So the concept of new infection agents entering human population and spreading rapidly around the world is not new. Before also, you can the very popular example is the plague. Plague was being carried by rodents, and rodents were these were the animals which were spreading plague. The plague came from these animals into humans, and human kept on spreading the virus. So it is actually a bacteria. I'm sorry. So plague is actually a bacteria which was carried by rodents. which later came into the humans. Similarly, HIV was present in the animals, the rhesus monkey, and then it came into humans. And this is the proximity of the humans to the animals actually lead to the transmission of the virus from the humans to the animals. So this is not a new thing which has happened. This is a very age-old thing which has happened. But this pandemic which we are facing is also has come from an animal. In this slide, you see that the primary host for this virus is a bat. Now, the primary host, the bat, doesn't itself get infected with this virus. You try to understand this. The bat doesn't itself gets infected with this virus, but it is acting as a reservoir for this virus, and it transmits the virus to the intermediate host, which could be civets in case of SARS-CoV, which was an earlier outbreak of a different SARS coronavirus, and the bat spread the virus to camels when we saw the MERS outbreak. But now the intermediate host has been spotted out to pangolins. Pangolins are small creatures, as you can see in this photograph. Pangolin soup is being shown here. That means pangolin soup is a famous delicacy in China, and pangolin was actually intermediate host, and the primary host was the bat. You can see in the photograph, bat is also a delicacy in China. So bat was the primary host, and the pangolin was the second intermediate host, which ultimately came into the humans by consuming or by the close proximity of these animals with the humans. So it was in the market of the animal market of the China where the humans came in close proximity to these animals, and the virus actually infected, or it changed the host from pangolins to humans, and later on the humans spread the virus to other humans, and it became a pandemic. Now let's talk about the vaccine development. I will tell you what are the different modes of making vaccines. Vaccines are represent one of the most effective and cost-effective medical and public health achievement of all times, because vaccines, they annually prevent about 3 million deaths. It is just due to the vaccination of human beings so many lives are saved across the globe. Now, vaccines are not made from a single strategy. There could be a number of strategies for making vaccines. As you can see here in the slide, it can be made by live attenuated virus, means you reduce the pathogenicity of the virus, and you can, after reducing the pathogenicity of the virus, it can be injected into a candidate human, which will generate immune response, and the immunoglobulins will be formed, which will save that person from a later attack from this disease. A vaccine can also be made by recombinant means. That means we will be expressing a specific antigenic protein of the virus, and that antigenic protein will trigger the immune response into a human being, and antibodies will be made in the human to fight against that virus. So that we can also have recombinant bacterial vectors, which will again produce antigenic protein that can be used as vaccine candidates. We can also generate viral like particles. These are small particles which are artificial viruses, but which do not infect, which do not replicate once they get inside the human. So they will just help in triggering the immune response. So these can also be used. Then we can also have recombinant viral vectors, which are equally good in triggering the response. We can have whole inactivated virus. We can chemically inactivate the virus, and then the virus can be given as a vaccine. So these are the different strategies which are available for the vaccine, for making the vaccine against this virus. Now, here, let me update you what is the latest going on in the field of vaccine when it comes to the COVID-19. So there are a number of vaccines which has entered into trials. In this picture, you see that the Serum Institute, which is there in the Pune, India, is the largest manufacturer of vaccines in volume. So 60 to 70% of the vaccines which are supplied across the world are actually supplied from Serum Institute, which is there in India. So in Serum Institute, a number of trials are the manufacturing facility of a number of firms are being carried out. So, as you know that this is a global pandemic, and Serum Institute is helping all these companies in manufacturing of these vaccines. So, let me tell you that there are about four vaccine candidates which have entered the phase 2 of the clinical trials. The number one is from the Oxford University. This is a very potential vaccine which has been made by Oxford University, and now AstraZeneca, a company, it has drawn an agreement with the Oxford University for manufacturing and global supply of this vaccine. So India is also one of the partners when it comes to manufacturing. So AstraZeneca has chosen the Serum Institute for manufacturing millions of doses of this vaccine. So the Serum Institute has started making this vaccine, but still the vaccine is in the stage 2 of the clinical trials. It cannot be marketed as of now. But just to tell you that United States has already booked a big consignment of vaccines from this company. The next company is Cadogenics, which is again a US firm which has collaborated with the Serum Institute for the manufacturing of these vaccines. The next is Novavax, again a US company, and then an Austria based company called Themis, which is again in collaboration with the Serum Institute of India. They are manufacturing big doses of these vaccines. Apart from Serum Institute of India, Cadila has also collaborated with US firms and also with other countries like Vietnam for manufacture of these vaccines. So I'm very sure that soon the good news will be coming for the successful trials of these vaccines. You should know that before coming into the market, the vaccine has to undergo 3 phases of clinical trials. Uh, First is the clinical trial phase 1, wherein about 40 to 50 people are being recruited. And once that is successful, the results are reviewed by FDA, and then the vaccine enters into the phase 2 of the trial, then the phase 3, which is being done on a larger group of people. So the phase 2 trials are being going on for this vaccine, and by the month of July, the vaccine will enter into the phase 3 trial, and we can expect a vaccine by the end of this year. So we are quite hopeful, and we are quite we look forward for receiving the doses of vaccines from with the manufacturing facility there in India itself. But the technology would be of the companies which are US based or Austria based or based in some other country. So wherever the technology is, but the manufacturing will definitely will happen in India. So in this picture, you see it is Mr. Poonawala, who is the owner of the Serum Institute of India. And sometime before, our Prime Minister Narendra Modi also visited this institute. So we are really proud of this institute because 60 to 70% of the vaccines which are sold all over the world comes from Serum Institute of India. So that's a matter of proud for all of us. And let's hope that the vaccine comes very soon out in the market. So I'll next take you to now, after finishing with the vaccine part, next two parts would be on the therapy and on the diagnosis. But before joining the therapy portion, I will tell you that there are certain concerns with the vaccines because we are not very sure that the vaccine, which is still under trial, is going to work. So there are some concerns which I will tell you in a short and in the coming slide. So after discussing the different candidates for the vaccines, which are primarily manufactured in Serum Institute of India, now I will tell you what are the concerns and what are the lacunae. Let me tell you very frankly that it is not every time possible that the vaccine becomes successful. So there are so many different candidate vaccines which are under trial. Many of these will be not that successful, and there are many vaccine candidates which will face one or the other problems. Now, what are those problems? Why are we not 100% sure that a vaccine is going to work in the coming future when we have a vaccine? So that's the reason the trials are going on, and uh, the candidate which is for safe and which develops all the immunity will be the successful candidate. Now, let me tell you what are the different concerns about the vaccine candidates. Now, in this slide, I'll show you the number one is that uh, what needs to be emphasized here is that vaccines are simple in principle, but they are complex in practice. Now, why do I say so? Is that uh, because, for example, a vaccine, if it is made by using adenovirus, another virus, we are putting the gene of coronavirus. Now, if a person has a pre-existing immunity by means of some other vaccine for adenovirus, then this vaccine will not be useful for that person. It won't trigger immune response in that person. And in normal day-to-day life, also we come across adenovirus. Sorry, if a person is previously infected with adenovirus, and if the vaccine is based on adenovirus, then due to the pre-existing immunity in the person, this person might not give a very good response to the vaccine. Second is, it has been found by research that coronavirus do not tend to trigger long-lasting immunity. That means the immunity is triggered, but is not triggered for a bigger period of time. It has been observed that it is the antibodies which they come down very quickly. So the coronavirus do not tend to trigger the long-lasting immunity. For example, if we take the example of the common cold, we all suffer from common cold. But why doesn't our body produces the immunity so that we don't get reinfected by the common cold? You all must be knowing that common cold is caused by a virus. So even after repeated exposure to that virus, why are we not able to develop immunity against this virus? So this is a question which is challenging the scientists. Now, coronavirus is also uh, giving some similar kind of reactions, like the immunity which is generated inside the body of a person, it doesn't last for longer. So that is something which is of big concern, right? So common colds are caused by human coronavirus, but the immune response fades so rapidly that people can become reinfected the next year. So, the ideal feature of a vaccine should be that it induces the immune response for a longer period of time, which is not happening in case of the coronaviruses. The immunoglobulin G antibodies, which is responsible for long-lasting immunity, it rose steeply initially when the person gets infected. The IgG antibody is goes higher, but in the after the first month of infection, it falls again. So that is something which is worrying the scientists. Why is the immunoglobulin G falling again? So if we vaccinate a person with a vaccine, initially the response of antibodies is good, but later on it fades out. That means now the person is prone to reinfection again. So that is a big concern, which is being addressed by the scientists. Scientists at Rockefeller University in New York found that most people who recovered from COVID 19 without going into hospital did not make any killer antibodies against the virus. So, once you are infected, you should be making those antibodies which can kill the virus when encounters later in our body. So this is not happening in case of the coronavirus. And another concern is that this virus, although it is very stable, but there are evidence that it is acquiring mutations. Now, if the virus gets mutated, you might lose the efficacy of the vaccine. If you are making vaccine against the spike protein of the virus, and later on the spike protein mutates, so your vaccine is ineffective. It is no more active again. So every year you will have to get vaccinated. So there is another concern that slowly the virus is acquiring some mutations, which is a big concern. Though the good thing is that this virus is quite stable till now, but there are evidence that it has acquired mutations. So that was about vaccine. Now we see what are the therapies which are available. But let me tell you, unfortunately, no medicine or an antiviral vaccine has yet been officially approved to treat COVID-19 associated pathologies. But still, there are certain broad spectrum antivirals, and there are certain known antivirals which are now being tried or which are being reproposed for the treatment of this virus. Now, how can we actually, the mechanism behind treating the coronavirus might be, if you look into this slide, it can either that if we use a monoclonal antibody from a convalescent plasma, that means from a person who is already being infected, or a monoclonal antibody which is against the spike protein, then this antibody will prevent the binding of the virus to the cell receptor. So you all must be knowing that the infection, in the process of infection, first the virus binds to the cell receptor, and then it is it goes inside the cell after binding to the cell receptor, and then hijacks the machinery of the cell and starts making its own replicas. And once the replicas are made, it is released by the rupture of the cell. So, when it comes to therapy, we can have either neutralizing antibodies or monoclonal antibodies which are against the spike protein of the virus, so that the virus is prevented from binding to the receptor, or we can have antibodies against the receptor itself, which will stop the binding of the virus to the receptor. The second line of therapy is that when the virus enters the cell and starts manufacturing its own copies of proteins, that proteins, if they can be lysed, or they, by means of certain proteases, some certain enzymes which will degrade these proteins, so the viral copies are not formed. So that is the second line of therapy. And the drug lopinavir and ritonavir are actually causing the proteolysis of the viral proteins, and thus which and it degrades or stops the spread of the virus or the replication of the virus. The third line of therapy is by stopping the replication, or by hindering the formation of the transcription complex. So the drug remdesivir is one which stops the replication of the virus by binding to the nucleic acids. RNA is the nucleic acid of this virus, and this virus is prevented from being getting replicated. Another two drugs which actually are meant for malarial treatment have been found very useful for the treatment of coronavirus. These are chloroquine and hydroxychloroquine. So these two drugs have also been showing very good results against coronavirus. Now let me tell you that these drugs are also in clinical trials, though they are known drugs, but they have again been put into clinical trials to test their efficacy against the coronavirus. So that was about the therapy. Now let me tell you about something about the diagnosis. So in diagnosis, there are two very famous tests which are being done. Although there are many other ways of diagnosing a coronavirus, which includes ELISA method, or even the CD scans, or even the blood test. But the most highly used and very quick methods are by RT-PCR. I won't say it is a quick method. It takes 6 to 7 hours for the results to come. But RT-PCR is one very good method of diagnosing a virus. Here you can see that the nasal swabs or the sputum swabs are taken, and this swab is then dipped into the virus carrying medium, and then it is taken to the lab where RNA is extracted from this virus. Then RNA, by means of the reverse transcriptase, is converted into a double-stranded DNA, and which is subjected to a PCR. And if this is called the RT-PCR, wherein the RNA is converted into DNA before amplifying the specified segment of the virus. So if at all the virus is present, you will find amplification of your nucleic acids. So this test interprets results. Presence of viral RNA indicates active SARS-CoV-2, that is COVID-19 infection. So this test is based on the method of PCR, and this is the test which is nowadays being used for the detection of this virus. There are certain problems with this test, that the RNA might get degraded while being carried to the lab, which is a big problem, or the virus sample gets degraded and its nucleic acid doesn't maintains its normal structure. So then false results can come. The other test which is being carried out is the antibody test, which detects the immune response of the COVID-19 exposure. If you are being exposed to COVID-19, your body will show the presence of the antibodies against this virus. So this test actually detects the antibody. Now here the sample is taken in the form of the blood of the patient, and the patient's blood is withdrawn, and you can see a slide on which antigens are being immobilized. These antigens react with the patient's antibody, and if antibodies are present against the COVID-19 antigen, which could be a spike protein antigen, or any other nucleocapsid protein antigen, which is being bound on this slide. And if if a patient is ever exposed to COVID-19, his blood will have those antibodies. And if the antibodies bind to the test line, a signal is being given in the form of a color reaction, which shows that the patient is exposed to COVID-19. The shortcoming is that even the patients who have recovered from this disease will again show a positive result because his body has some antibodies against the COVID-19. So this is a rapid test, but it is not very sure shot. So that was all about the COVID-19. I thank you all. In the picture, you see my department, that is Department of Microbiology at Maharshi University, Rohtak. I thank you all and for your time, and I wish you super success for the coming lectures uh, in the series of the FTP. Thank you so much.